Hello everyone, I am Professor Angshuman Bagchi, Department of Biochemistry and Biophysics, University of Kalyani. I am also the coordinator of the DBT funded Bioinformatics Infrastructure Facility at University of Kalyani. Today, I'll be talking about my work on non-coding RNAs, specifically the long non-coding RNAs and the circular RNAs and their protein products. So, as the name implies, the long non-coding RNAs are circular RNAs. These are the RNA molecules which are not associated with the coding of proteins. How are they synthesized? Their synthesis occurs in the nucleus from the heteronuclear RNA, and the synthesis process is a very complicated one. What happens is during the synthesis, there are joining of exons and the intron regions. Now, as the name implies, these long non-coding RNAs and the circular RNAs, they belong to the category of non-coding RNAs. Still, some of them have distinct protein coding potentials. The long non-coding RNAs, which can code for proteins, they have an intronic region which contains a small ORF, with the help of which they can synthesize proteins. For circular RNAs, there are specifically two different mechanisms. One is the presence of internal ribosomal entry sites to which ribosomes can bind and initiate the further protein synthesis process. Also, there are certain M6A modifications. Those M6A modifications, what they do is they help in binding the ribosomes, attracting the ribosomes, and the protein synthesis may take place. Today, I will be talking about a protein named MAPK1109AA, which is coming from SERC MAPK1. Now, why we are interested in this protein MAPK1109AA? That is because it is known that this particular protein MAPK1109AA is an inhibitor of gastric cancer pathways. Now, what happens during gastric cancer onset? In our cells, we have the regular MAPK1 protein, which gets phosphorylated by MEK1, and after being phosphorylated, the protein initiates the downstream transcription processes for the onset of gastric cancer. Now, whenever MAPK1 109AA, this particular protein obtained from the circular RNA SARC-MAPK1, when this protein is present along with MAPK1, the second one, that means the MAPK1 109AA, kind of competes with MAPK1 to bind to MEK1. And what we observe is that MAPK1 109AA binds in a better way with MEK1, so that the MAPK1 109AA gets phosphorylated and thereby activated, and MAPK1 is not activated. That means the phosphorylation event is not there. And when the MAPK1 109AA is present in its active form, what happens is the downstream processes necessary for the onset of gastric cancer, those processes are prevented. Now, till date, there are no structures of this MAPK1 109AA. So we used bioinformatics techniques to generate structures, and what you observed is that this is an alpha beta protein and its active site residues, that means there is a tyrosine and threonine residues which get phosphorylated by MEK1. These active site residues are present in a flexible loop region so that they can easily interact with its binding partner. Now, we observe the binding interactions of MAPK1 109AA with MEK1 and MAPK1 with MEK1, and what you observe is that MAPK1 109AA binds in a better way with MEK1 as compared to MAPK1 109AA binds in a better way with MEK1 as compared to MAPK1. That means when MAPK1 and MAPK1 109AA, both these proteins are present, there is a competitive interaction, and MAPK1 109AA generally wins. When we further studied its binding interactions, what we observed that MAPK1 109AA has its ability to bind to different other anti-proliferative ligands, and this binding would help in further activating its function in the gastric cancer pathway. Furthermore, we observed that there are certain known inhibitors of gastric cancer which bind very well with MAPK1. But those ligands, those inhibitors of gastric cancer, bind to MAPK1 109AA in a region so that the functionality of MAPK1 109AA is not at all hampered. In other words, when MAPK1 and MAPK1 109AA, both of them are present in a patient having gastric cancer, and when that patient is treated with all those inhibitory ligands, what happens is those ligands can inhibit the activity of MAPK1 but cannot inhibit the activity of MAPK1 109AA. Furthermore, when we analyze the further binding interactions of MAPK1 109AA, we observe that not only gastric cancer but MAPK1 109AA is also associated with the other types of regulatory processes, and one of the most important ones among them is the neurotrophin signaling pathway. Therefore, what we can conclude here is that MAPK1 109AA, which is coming from the circular RNA SARC-MAPK1, can be considered as an alternative therapy for the treatment of gastric cancer. This work was published in Journal of Molecular Graphics and Modeling, and with this I have come to the end of my presentation, and I would like to thank the organizers of this event for giving me this opportunity to present my work, as well as my heartfelt thanks goes to the Department of Biotechnology, Government of India for the funding. I am also thankful to ICMR for the funding. Thank you very much.
Namaskar, all dear delegates, colleagues, all the bureaucrats, all the scientists in this online conference. And I congratulate Vedant Group of Systems, Vedant Knowledge Systems, for organizing such a nice series of my Viksit Bharat conferences, where each researcher gets a nice opportunity to present his or her research work to the entire fraternity, including of his or her subject and many other subjects. The themes selected are quite diversified so that each researcher gets a chance. I myself, Dr. Mehul Dave, and I'm working as an associate professor and head at Department of Life Science at IITE, Gandhinagar, Gujarat, India. IITE stands for Indian Institute of Teacher Education. This is the only university, teacher education university in entire Southeast Asia. And it's a dream child of our beloved Prime Minister Pradhan Mantri Modi ji. When he was CM, he established this university where I work. I had the opportunity to chair the session where total 7 papers are presented, and these 7 papers are also, in various aspects, they are quite diversified in the sub-study of life sciences. The papers, they range from DNA damages and non-coding RNA to altogether to sustainable development goals and Indian knowledge system. Some of the papers are in the subject of botany and medical science and diagnosis also, and some of the modern biotechnological research also. So again, congratulating all. I will present the review of the first paper, that is of Dr. Angshuman Bagchi, and the title of the paper is Can the Non-Coding RNA Become Coding? So this paper is given in a video format where there are no slides. The paper addresses a fundamental shift in our understanding of the genome by demonstrating that certain non-coding RNAs may also encode functional proteins. So this is a good concept explored. The study effectively uses computational biology to predict molecular features and cellular dynamics of these RNA-derived products. Importantly, the work of Dr. Anshuman Bagchi, it presents the challenges and it gives a challenge to the long-standing assumption in our mind about the gene regulation and opens some new questions about functional annotation of the human genome. So this paper is, from that point of view, it is good paper. The implications for disease mechanisms, especially in cancer and neurodegradation, makes this is a impactful contribution. So altogether, the paper explains a fundamental shift in our thoughts that non-coding RNA can never have function. But here it has been tried to explore that this non-coding RNA can also become coding. So we wish that more research is done and the paper is presented with more data to justify the title. So I congratulate Dr. Angshuman Bagchi for presenting a good paper. Thank you.
Good afternoon, everyone. Myself, Dr. Chhanda Mallick, Mukherjee, Assistant Professor, Department of Biomedical Laboratory Science and Management, Vidyasagar University, Midnapur, West Bengal. First of all, I like to thank Dr. Arjun Dave, the Director of Vedant Knowledge System, for providing me such an international platform to deliver my talk. Today, I will talk about the comprehensive strategy to combat anemia, global and Indian perspective and linkage with the Sustainable Development Goal. Anemia remains one of the most pervasive yet preventable health challenges globally. Despite decades of intervention, it continues to affect nearly 2 billion people worldwide, with the greatest burden borne by women of reproductive age, children, and adolescents. Anemia is not merely the medical condition, it is a reflection of broader nutritional, social, and economic inequities that directly hinder sustainable development. Globally, nearly 1 in 3 women and about 40% of children under 5 years are anemic. The consequences are profound, ranging from impaired cognitive and physical development in children to reduced work capacity, adverse pregnancy outcome, and increased maternal mortality. These impacts extend beyond health, affecting education, productivity, national economic growth. India carries a disproportionately high share of global anemic burden. National data revealed that anemia affects more than half of the women of reproductive age, nearly two-thirds of young children, and significant proportion of adolescents. Persistently high prevalence reflects poor dietary diversity, micronutrient deficiency, recurrent infection, early marriage, frequent pregnancies, and socio-economic disparities. Recognizing this, the Government of India launched Anemia Mukt Bharat program in 2018 under the Poshan Abhiyan. It's a life cycle approach targeting 6 population groups from early childhood to adulthood. Its core strategy include iron folic acid supplementation, periodic deworming, promotion of the fortified and iron-rich food, point of care anemia testing, behavior change communication, and addressing non-nutritional causes such as infection and hemoglobinopathies. However, several implementation challenges remain. These include supply chain disruption, poor adherence due to side effect and misconception, limited focus on dietary diversification, inadequate screening for non-iron deficiency anemia, and gap in monitoring and evaluation. These limitations highlight the need for more refined, data-driven, and people-centered approach. A new roadmap for anemia control must move beyond supplementation alone. It should emphasize precision public health through improved screening and digital monitoring, strengthen food fortification, promotion of locally available iron-rich diet, integration of infection control, and enhance community engagement. Health system strengthening and capacity building at the grassroots level are also very crucial. Importantly, combating anemia is directly aligned with Sustainable Development Goals. It contributes to SDG 2, that is the zero hunger, by addressing malnutrition and micronutrient deficiency, and SDG 3, that is the good health and well-being, by reducing maternal and child mortality and morbidity. Improved iron status, enhanced learning and productivity, supporting the SDG 4, that is quality education, and SDG 8, that is the decent work and economic growth, by improving the health of women, adolescent girl, anemia reduction, advance the gender equality. Collectively, these gains help break the cycle of poverty, contributing to SDG 1, that is the no poverty. In conclusion, anemia is not merely a health indicator, but a critical marker of sustainable development. A comprehensive multi-sectoral strategy globally and in India is very essential, that is rooted in equity, innovation, and community participation. By prioritizing the anemia control, we invest in healthier population, stronger economics, and more sustainable future. Thank you.
Namaste to all the delegates and researchers. I congratulate Vedant Knowledge Systems for organizing this conference, and among the various papers presented here in online mode, I had got the opportunity to chair a session of reviewing the life science related papers, and now I'm reviewing and giving my feedback on the paper of Dr. Chhanda Mallick Mukherjee. The title of the paper is Comprehensive Strategy to Combat Anemia, Global and Indian Perspectives and Linkage with Sustainable Development Goals. This paper represents a clear data-driven analysis of anemia as a persistent public health challenge. It has been explained nicely that anemia is prevalent in our population, and we have a number of different remedies for combating this anemia. But here the strong focus is given on the Indian context, and the author successfully integrates epidemiological evidence with national programs such as Anemia Mukta Bharat. It has been nicely addressed and explained. Global SDG commitments also are covered up in the paper. The emphasis on a life cycle and multi-sectorial approach highlights the gaps between policy intent and ground level implementation. So this has been addressed well. Overall, the paper provides valuable insights for researchers, policymakers, and public health practitioners alike. So this paper, Comprehensive Strategy to Combat Anemia, is a good paper, but this paper is presented without any slides and without any research data. So otherwise, the quality of the paper is good. Thank you.
Namaskar, respected dignitaries of Vedant Knowledge System, esteemed academicians and distinguished participants. On behalf of all my co-author, Dr. Rahul S. Gohel, Dr. Nehati Patel, research scholar Riddhi Sakrelia, and I, Dr. Reena P. Head and Associate Professor, Department of Biology, Shri M.N. Virani Science College, I am honored to present my review article titled Integrating Endogenous Bharatiya Wisdom, Environment Conservation and Sustainable Development Goal. Biology is the science of life, teaches us that every organism is indirectly connected and environment truly delicate ecological relationship. The principle of energy flow, nutrient cycle, biodiversity conservation, and ecological balance form the foundation of the environmental sustainability, integrity, And this scientific concept aligns deeply with the ancient Bharatiya wisdom and always emphasizes the harmony between humans and nature. This paper reviewed, I highlight how the age-old ancient wisdom, ancient philosophy or Indian philosophy rooted in Vedas, Upanishads and Mahatma's thoughts mirror modern technologies, biological principles, concept with ahimsa, aparigraha, sarvodaya and dharma, advocate ecological ethics and that support biodiversity, sustainable resources, uses and intergenerationally equity ideas and restore strongly modern sustainable development goals. Biology shows the ecosystem thrive on balance, diversity and minimal exploitations, and the Bharatiya wisdom promote exactly this value through practices like organic farming, sacred groves, water harvesting system, Ayurveda and yoga with a healthy, holistic lifestyle, all of which are biologically sustainable. This traditional supports SDGs sustainable development goals related to health, water, climate actions and agriculture and life on lands. So this review article paper explore how Bharatiya wisdom rooted with the ancient texts and sustainable goal. So these are the objectives of the study: sustainable development goal and interaction between the sustainable goals and their solutions. So Bharatiya wisdom and SDGs goal, the collective approach, the sustainable goal 2, zero hunger, sustainable goal 3, good health and well-being. These are some activities with the students. They're making some valuable agricultural and organic materials matter. These are the quality education we through the VACA course, value-added course. SDGs goal 6, clean water and sanitization. SDGs goal 12, responsible consumptions and productions. SDG goal 13, climate actions, and with the plantations of the students and some Smrutivan at the place of Rajkot, Ajden. SDGs goal 15, life on land, with the forest conservation. We all together observe how the forest conserve and the area, and these are some pictures of the Sasangir study 2. So this review article reviews the ancient Indian practices, emphasize living the harmony with the nature, traditional water management, agriculture practices and agricultural assignments, and some tailored local ecosystem, ensure resources efficiency and ecological balances. So I conclude the Bharatiya wisdom with some management practices and how it relates with some vital resources addressing contemporary global challenges, achieving the sustainable development goal. I acknowledge the dignitaries of Vedanta knowledge system. I acknowledge so for the providing the great platform, and I also thankful to Dr. K.D. Ladwa sir, and also thankful to all dignitaries of Atma University for their best and valuable supports. Thank you.
Namaskar to all the delegates and researchers. This is a part of the conference organized by Vedant Knowledge Systems, where I got the opportunity to chair the session. The paper that I am now giving feedback on and putting my review is of the researchers and Professor Dr. Reena Dave and team. The title of the paper is Integrating Indigenous Bharatiya Wisdom with Environment Conservation and Sustainable Development Goal. The paper is perfectly written and very well presented with all the necessary components for a scientific presentation. The author has included each aspect of a good research presentation, the paper offers rich interdisciplinary perspectives by linking ancient Bharatiya wisdom with modern sustainability science and sustainable development goals. Various SDGs are addressed here in this paper. The authors convincingly show how traditional ecological knowledge contributes to environmental conservation, health systems, even agriculture and education also. The work stands out for its philosophical depth, supported by scientific literature and real-world example. Dr. Reena Dave and the team has taken advantage of the teaching profession and motivated students to understand SDGs in the form of experiential learning and practical aspects and activities. It provides a strong conceptual framework for integrating indigenous knowledge systems into contemporary idea of sustainability and policies to education also. So overall, the paper is really good. It addresses various sustainable development goals like SDG number 3, 6, 12, 15. They are very well addressed in this research paper. So congratulations to the team of Dr. Reena Dave and the researchers. Thank you.
Good morning, everyone. I'm Dr. Umakanta Swain from Department of Zoology, School of Biological Science at AIPG University, Bhubaneswar. Thank you, organizers, for giving me opportunity to present our work. So today I will be presenting our work entitled The Non-Canonical Polymerase, that is 10-4A, governs DNA damage tolerance through mRNA stabilizations and non-coding role of how it is regulated the DNA damage response. That is a newly discovered long non-coding RNA, that is LNC RNA, PAXP1AS2. So what are the challenges of the replication stress? So to start, we know that for a cell to survive replication stress, it must effectively manage stalled replication fork. And this process relies heavily on timely requirement of specific enzymes, Y family DNA polymerase. However, the regulatory mechanisms ensuring this polymerase are available when needed are complex. So our study reports a novel post-transcriptional control mechanism for translation DNA synthesis, or TLS, mediated by non-canonical polymer 10-4A, also known as PAPD7s. So what is the essential role of this 10-4A? So we discovered that DNA4A is absolutely essential for cell survival following the DNA damage. So mainly it functions as in context to maintain the intracellular pool of two important critical protein, that is TLS DNA polymerase eta and RAD18, which is a PCNA ubiquitinating ligase. So without 10-4A, the cell failed to maintain the adequate level of this factor. So overall, if we see the mechanism we found, this is due to mRNA stabilization by polyadenylation. So our analysis revealed 10-4A appropriately by directly regulating the stability of translations of polyeta and RAD18 transcripts. So it achieved through like a non-canonical way of the polyadenylation. So that means it add short poly A tail or maybe other nucleotides. Essentially, the loss of this 10-4A results in destabilizations of this specific mRNA. Consequently, this prevents the assembly of the function of TLS complex and stalled replication fork, compromising the cell's ability to repair the damage. In sum up, if we sum up the whole work, that is the regulatory network and role of long non-coding RNA. However, this is going beyond a simple way of the mRNA regulation. So we found that 10-4A sits at the apex of the complex signaling network, and this network involves a D-ubiquitinase called as SHIELD, which is a crucial non-coding RNA, that is PAXP1AS2. We have identified that PAXP1AS2 is a novel repressor of RAD18 expression. When that 10-4A is lost, this non-coding RNA is upregulated, which contribute directly to the TLS effect. So conversely, we observe that shield is required for the stabilizations for the RAD18. So in summary, so our data defines a specific role of tent for a dependent RNA regulatory circuit, and this circuit fine tunes the DNA damage tolerance capacity to the human cells. Ultimately, this work highlights a critical link between RNA processing and maintaining the genomic stability. With this, I'm concluding my short talk, and thank you very much for your attentions. Thank you.
Namaste to all the delegates joining in Viksit Bharat conference organized by Vedant Knowledge Systems, and I congratulate Vedant Knowledge Systems for organizing such a nice online conference where every researcher gets some opportunity to present his or her work. Now I'm getting the opportunity to give my feedback and reviews on the paper written by Dr. Umakanta Swain. The title of the paper and the concept of the paper is really good. The role of TENT4A in DNA damage tolerance via RNA regulation. So this is a good molecular biology research work, and it aims to solve some of the fundamental problems that we have in molecular biology, gene expression, gene regulation also. This is a rigorous molecular biology study, as I told, and it uncovers a novel RNA-based regulatory circuit governing DNA damage tolerance. So we know various DNA repair mechanisms like SOS system and photoreactivation and mismatch repair and recombinational repair, excision repair. But now after this research, the TENT-RA will be studied more by the researchers. The identification of TENT4A, it's a key post-transcriptional regulator that adds significant and mechanistic depth to our understanding of genome stability. The work also highlights the role of INC-RNA, that is in fine-tuning the DNA repair pathways. So new aspects in the DNA repair mechanisms are explained here in this paper. The findings will have a strong relevance for cancer biology and future therapeutic research also. This paper is presented without any slides and without the research data. That can be done further for strengthening the concept and for appropriate justification of all the conclusions which are derived by the researchers. Otherwise, the quality and the subject and the title of the paper is really good. Some of the research components in this paper would have been added, then it would be more scientifically correct paper. Thank you. Thank you very much.
Good morning to all. My study, Role of Botanical Education in Building a Herbal-Based Rural Economy for Viksit Bharat 2047, highlights how scientific plant knowledge can empower rural communities. India has huge medicinal plant diversity, but rural population often lacks scientific training in plant identification, cultivation, harvesting, and value addition. Botanical education fills this gap. Case studies from Uttarakhand, Chhattisgarh, and Assam. The research shows that can SHGs and youth receive proper botanical training, rural income rises by 30% to 400%. Women IED SHGs become successful herbal entrepreneurs. Sustainable harvesting improves quality and protects biodiversity. MSMEs grow due to standardized high quality raw materials. This education transforms villages into hubs of herbal processing, product diversification, and entrepreneurship. The study recommends establishing herbal skill centers, certification courses, processing clusters, and digital training platforms to strengthen the herbal economy. In conclusion, botanical education is a powerful driver of rural empowerment, women leadership, and sustainable economic growth essential for achieving Viksit Bharat 2047. Thank you.
Namaskar to all the research delegates and all the participants in this conference. Namaskar to all the scholars, professors, academicians, and all the technocrats also. Now I'm giving my feedback and review on the paper presented by Galsor, Bhagvatiben, Jivabhai in this My Viksit Bharat conference. This is paper as a title. The researcher has selected role of botanical education in building a herbal-based rural economy for Viksit Bharat. So this paper is very well relevant to the theme of the conference, which was suggested by Vedant Systems. This paper clearly demonstrates how botanical education can drive sustainable rural livelihoods and economic empowerment. The focus on SHGs, women leadership, and MSME development. It strengthens the social and developmental relations of the study. The paper is presented without any research data and no PowerPoint or no other slides are presented. By combining policy analysis with regional case studies, the work offers practical scalable models for herbal entrepreneurship. So this is a good part, and this can lead to a good startup in this subject. The paper aligns well with national priorities under Viksit Bharat Vision and Green Economy initiatives. Researchers Galsor Bhagvatiben has tried to initiate the research with a very nice subject line, but the more profound research data and practical aspects would justify the strength of the research which is carried out. So that should be added in this paper. Thank you.
Sustainable Development Goals 2030 Bridging Gaps Building Futures for Viksit Bharat at 2047. I'm A. Melzika Manuel, a student, and I'm one of the co-authors in this research paper. We are from Christ Kingeri campus, and today I'm here to present the paper titled A Statistical Study on the Effect of the Solvent and Binder on the Particle Size of ZNO-based inks for Additive Manufacturing. This work was carried out by the Department of Electronics and Communication Engineering, Christ University. The study focuses on understanding how solvent composition and binder concentration affect particle size and stability, which are critical for printable functional inks. From existing literature, zinc oxide is widely studied for printed electronic sensors and optoelectronic applications because of its good electrical and optical properties. Previous studies show that nanoparticle inks require controlled particle size and stability. Solvent polarity is known to influence particle agglomeration, while polymer binders affect dispersion and film formation. Dynamic light scattering is commonly used to study particle size, PDI, and diffusion behavior. However, most studies focus on either solvent effects or binder effects individually. There are limited studies that analyze the combined effect of solvent composition and binder concentration, especially from a printability point of view. This gap forms the basis of our work. In general, nanoparticle inks are formulated using approaches such as solvent-based stabilization, surfactant-assisted dispersion, polymer binder addition, and pH control. While these methods improve dispersion and film quality, they also introduce challenges like aggregation, viscosity mismatch, and nozzle clogging if not properly optimized. Hence, ink formulation involves balancing stability with print compatibility. Directing writing is an extrusion-based additive manufacturing technique where ink is pushed through a micron-scale nozzle. It is suitable for printing thick films and functional patterns, but it is highly sensitive to particle size and rheology. Poorly dispersed or large particles can easily cause nozzle clogging, highlighting the importance of particle size control in the ink formulation. The main problem addressed in this study is that the zinc oxide nanoparticles tend to agglomerate in liquid inks. This agglomeration leads to nozzle clogging, poor print resolution, and inconsistent device performance. While nanoparticles are desirable, printing systems impose strict particle size limits. There is a lack of quantitative framework linking solvent composition and binder concentration with particle size and diffusion. Our work aims to address this issue. Zinc oxide is a key material for printed electronics, but its tendency to agglomerate make ink formulation challenging. The objectives of this work are to study the effect of water-ethanol solvent ratio on ZNO ink stability, analyze the role of PVA binder in particle dispersion and agglomeration, quantify ink properties using dynamic light scattering, identify ink formulations suitable for additive manufacturing constraints. ZNO nanoparticles were synthesized using the co-precipitation method. Ink formulations were prepared using different water-ethanol solvent ratios, with PVA added as a binder in selected samples. Here are the input parameters of the sample. The synthesis conditions were controlled with pH maintained between 8 and 10, temperature around 60 degrees Celsius, and continuous stirring. All samples were characterized using dynamic light scattering. DLS results showed that all zinc Adeno inks exhibit multimodal particle size distributions. Two main size populations were observed: 100 to 150 nanometers, corresponding to nanoparticles or small clusters, 1 to 4 micrometers, corresponding to agglomerated particles. Here are the graphs for the same. The solvent composition had a strong influence on particle size. Water-rich systems produce smaller particle sizes and higher diffusion coefficients, while ethanol-rich systems promote agglomeration. The addition of PBA at low concentration resulted in uniform micron-size agglomerates and reduced PTI. This behavior is attributed to bridging flocculation, where polymer chains connect multiple particles. Statistical correlation analysis confirmed that solvent polarity is a dominant factor, while binder concentration has a weaker influence at the tested levels. To summarize the key findings, Zedeno inks show multimodal size distributions, water-rich solvents yield smaller particle sizes, ethanol promotes agglomeration, low PVA concentration leads to uniform micron size agglomerates. Importantly, the particle sizes obtained fall within the printable range of approximately 1 to 2 micrometers, which is suitable for additive manufacturing. In conclusion, solvent composition is a primary factor controlling particle size, and this study provides a practical framework for Zedeno ink formulation. The developed Zedeno inks are suitable for applications such as printed sensors, transparent conducting layers, flexible electronics, and optoelectronic devices. Future work includes optimizing binder concentration, exploring alternative surfactants, and performing rheological studies, and also testing printed devices. Here are the references. Thank you for your attention.
Namaskar to all the research delegates and participants of the conference. Namaskar to all the senior dignitaries in this Vedant Knowledge System conference, which is organized nicely in online mode. And the theme which has been given to me for review is life sciences. And in the life sciences, there are 7 papers, out of which some of the papers are in subject of sustainable development goals. Some of them are addressing molecular biology problems, and some of them are true biotechnological, botany, and medical science related papers also. So here now I'm giving my review on the paper, which is prepared by and presented by SOURAV Roy and team. And this paper title is Statistical Study on Solvent and Binder Effect in Zinc Oxide Ink for Additive Manufacturing. So the concept and the title and the research area problem statement selected by the researcher is really good. It's a modern biotechnology and application based research work. So I congratulate authors for selecting a nice topic. The paper presents a well-structured experimental and statistical investigation of zinc oxide-based ink formulations for additive manufacturing. This nanoparticle inks, they require controlled particle size and stability. And the problem which is addressed in this research paper is agglomeration of the liquid ink. And here the solution of preventing this agglomeration are explained well by the researchers by giving appropriate slides and research data also. So this paper which is presented, it covers all the necessary aspects of a good research presentation. So I congratulate them. So how agglomeration must be avoided, that is very well explained by them. The integration of dynamic light scattering with regression and ANOVA analysis enhances the robustness of the finding of the researcher. The study provides actionable insights into controlling particle size and dispersion stability, which are critical for printability and use of an application of any good ink. The objectives are very well addressed. Good objectives are also selected by the researcher. The work is particularly relevant for advancing the printed electronic sensor and functional material, and various applications and future works mentioned in the paper, they are also good. So as such, zinc oxide is widely used, widely chosen for printed electronic and sensors. And here the novel concepts and preventing agglomeration of this type of nanoparticle based ink will really add to the future development in this area. So it's a good research presentation. So I congratulate Mr. Saurav Roy and the team. Thank you.
Hello everyone, this is Deepika Vadivelu from Chennai, Tamil Nadu. I am an aspiring PhD research scholar at Wales University in Chennai. I sincerely thank the team Vedant for giving me this wonderful opportunity. Thank you. So, the title of my review paper is Phytochemical Diversity and Anti-Cancer Mechanism of Drinaria quasifolia. Trinaria quasifolia, locally known as Mudavatukal kilangu, is a traditional medicinal fern that has been used in Indian ethnomedicine for many years. It is commonly used for treating bone disorders, inflammation, wound healing, and gastrointestinal problems. In recent years, cancer has become one of the most prevalent diseases worldwide. Although treatments such as chemotherapy are available, they are often associated with severe side effects. Because of this, there is increasing interest in plant based natural compounds as safer anti cancer alternatives. Several published studies have reported that Drinaria quasifolia contains a wide range of bioactive phytochemicals, including flavonoids, phenolic acid, and terpenoids. These compounds have been identified using advanced analytical techniques such as GCMS gas chromatography method and HRLCMS and UPLCMS. In vitro studies reported in the literature have been demonstrated significant anti-cancer activity of Trinaria quasifolia. Extracts against various cancer cell lines, including lung cancer, breast cancer, and osteosarcoma. These studies indicate notable cytotoxic and anti-proliferative effects. Mechanistic evidence suggests that these anti-cancer effects are mainly mediated through apoptosis and involving ROS, that is reactive oxygen species modulation, mitochondrial membrane potential disruption, and cat space activation. So, despite these promising findings, there are still several research gaps out there, such as limited isolation of active compounds, incomplete understanding of molecular mechanisms, restricted cancer models, and lack of in vivo and clinical studies. Coming to the conclusion part, Drinaria quasifolia shows a very strong potential as a phytochemical source for anti-cancer drug discovery. However, further detailed mechanistic and translation studies are required to fully explore its therapeutic value. So, thank you so much for giving this opportunity and thank you for your kind attention. Thank you.
Namaskar to all the delegates and participants in this Viksit Bharat conference, which is organized by Vedant Knowledge Systems. Myself, Dr. Mehul Dave, and I will be giving review on the paper presented by Vadivelu Deepika and Bhupati Usharani. The title of the paper is Elucidating the Phytochemical Diversity and Anti-Cancer Mechanisms of Drinaria quercifolia. So this review paper, it's a review paper. It is presented without any slides. This review paper brings together extensive phytochemical and in vitro cancer evidence on Drinaria quercifolia. It's a traditionally important medicinal fern. So good medicinal property of this fern plant is explained in this review paper. The author systematically discussed the cytotoxicity, apoptosis, and cell cycle regulation while maintaining a critical scientific perspective and anti-cancer activities and mechanisms. If this Drinaria quercifolia proves to be anti-cancer material, then it is of great medicinal value, as we know. Notably, the paper identifies clear gaps in mechanistic understanding and translational research. It lays strong foundation for future drug discovery and phytomedicine research. This paper chosen good area of research, but more data is needed to justify the findings which are explained in this research paper. But I congratulate Vadivelu Deepika and Bhupati Usharani for selecting such a nice title and topic, and I will recommend them to carry out further more active research in this field, which will give some benefit to the future mankind. Thank you very much.